Good afternoon, everyone. I'm very glad to present here on behalf of our genomic team. So I'm gonna introduce a little bit our past work regarding genetic and genomics, and I hope you would find this is very interesting and would join us to move this forward. So in the clinical genetics and genomics, we have two staffs working here. One is Professor Dr. Josephine Zhong, and another one is me. So in our team, the major research interest is gonna working on translating genomic research into clinical advance for patients to improve the precise diagnosis, risk stratification, and personalized treatment. Currently, we have several projects ongoing. First is a genomic identification and functional characterization of disease-causing genes. So I show you one of our publication on this type of topic. So we kind of using the high throughput next generation sequencing like whole exome sequencing or genome sequencing to sequencing a group of a patient with similar presentations or in a special clinic. And with the data analysis, we're going to find out whether we're going to looking for pathogenic variants to explain the phenotype of this group of patients. And very luckily, we find a new gene that has never reported before that can help us to explain this new cohort of these a new type of diseases. So all the patients kind of have multiple congenital abnormalities with different degree of severities, and they are all caused by the biallelic mutation on the same gene. So for all our studies kind of combine a wet lab and the dry lab. So it's recruiting, we're gonna kind of have working on to improve the wet lab to see use less DNA or a new type of technology to improve the sequencing performance. At the same time, we're gonna work with bioinformatics team to help us to analysis the genome sequencing data. So here show so for in this study we are identifying new gene that to explain the patient disease and on the NAD de novo synthesis pathway, and also expand the genotype spectrum of this type of disorders. And the second part, we because currently the not only in the pediatric but also in other departments, it's kind of very popular to apply various genetic testing to find out the cause of the diseases. So identifying the genetic information is very important for the patients and also their family members. It's help to counseling and the family members was the recurrent risk of the future pregnancy to help predict prognosis of the diseases. So in this cohort study, we applied microarray. It's kind of currently is a more mature technology. So apply this microarray on a prenatal cohort of oral clefts. We find in this study we find compare with old technology like karyotyping or other karyotyping, we find 15% of the prenatal oral cleft was actually identified by the pathogenic CMV. So this kind of missed by those traditional relatively low resolution technologies. And for those, sorry, this type here, it's for those non-syndromic, which means only present with the prenatal oral cleft cases, they have a lower chance to find the pathogenic, while for those syndromic cases, you have a higher chance to find pathogenic variants. And also for those with non-syndromic cases plus some soft markers, which means for in the prenatal ultrasound, it's not a congenital abnormalities, more like polymorphism or variation that can be detected in the prenatal ultrasound. And for this group cohort, it's high. It has 20% of patients gonna find abnormal pathogenic CMV. So this is quite impressive because compared with non-syndromic, we never considered soft marker would play such a significant impact. And also because we have the genomic data and we kind of go for a deeper analysis correlate with genotype and phenotype, we kind of prioritize several candidate genes that may have a defect deleterious impact on the oral cleft or related to the orofacial development. And we also apply whole genome sequencing into the a cohort of pediatric patients. So currently we major focusing on with the patients with autism, multiple congenital abnormalities, development delay, this type of disorders. So we use 30 times whole genome sequencing and to sequence the trio or the proband only. So with this whole genome sequence data, we will generate 3 types of data. One is copy number changes, another is structural variation, including translocation, inversion, insertion, this type of very rare structural rearrangement, and also single nucleotide changes. So in general, for each case, we have 4 million point mutations, 10 kind of structural variations, and 100 copy number changes. And then, because this is a very large data set, it is impossible to analyze one by one. So you need to come up with a pipeline, a workflow to help you filtering out those common ones, those rare ones, and prioritize some interesting variants for your analysis. So we're gonna introduce different type of analysis pipelines, and this pipelines is kind of upgrading time over time because of the more knowledge regarding this field, and also some increased database and new database as well. So for each case, we're gonna have around 10 candidates variants, so help you to do a more depth correlation and analysis. So this is a general workflow for the sequence data analysis. We're gonna do the wet lab by ourselves, and also majority of them gonna be in-house pipeline, and then this part of it would be more manual work. So it's kind of a lot of cross-check and paper checkup. And for currently, we have some guidelines help you to help you to analyze this data. Because prioritizing this pathogenic variants is a very time-consuming and labor-intensive work, but you need to know some general frames that can be used and follow. So for the same group of data, different people have their own view, but for the very core part, the pathogenic part is going to be consistent among different labs. And so currently, the majority of the variants actually focusing on the coding region of the sequencing data, such as all the exons that can be translated into part of the protein. But actually, right now, we are more using whole genome sequencing data, so we're going to see the variants show up everywhere. And besides the coding region, actually deep intronic variant is also very currently is showing its play an important role in the human disease. We know it was there, but we don't have the power or the knowledge to analyze it. Currently, we are also focusing on how to interpret or build a model to help us to pick up those deep intronic variants. So we kind of have a patient find we also find a pathogenic variant from inherited from mother, and this is another deep intronic variant inherited from father. But how are we going to prove this? We will use mini-gene assay to insert a fragment of these proteins, include the fragment of DNA segment from the patients to the PCR to amplify this DNA fragment. And in this fragment, it includes the pathogenic variant. So with this technology, we're going to see how this deep intronic variant have an impact on the splicing. So we're going to do the RT-PCR to extract the RNA and do the RT-PCR to see what's the transcript of this target region. So you can see here, we will show there's 2 different types of transcripts that caused by this splicing site. One is include a part of the intron or retaining a new part intron next to the exon. So it's kind of causing the wrong signal, showing the wrong signal of the splicing. Yeah. So also kind of So in summary here, we are major doing a genetic investigation of the pediatric patients that's suspected with various genetic disorders, or consider or those patients are considered with which genetic role play a very significant part. And also we're doing some services to do regarding the prenatal diagnosis and also prenatal genetic diagnosis as well. Thank you.